Our protocol presents two assays quantifying C. elegans movement. These methods evaluate motility phenotypes, such as those exhibited by models of amyotrophic lateral sclerosis, or ALS. The radiolocomotion assay is a cost-effective and easy method to detect crawling on a solid surface. The swimming assay uses computer-based tracking for unbiased detection of thrashing movements in liquid. These methods are useful for quantifying movement differences in C. elegans. Although we study ALS, they can be used for any strain with altered motility.
Flip the NGM assay plates upside down and label the bottom with an identifier for the C. elegans strains to be assayed. Make a small dot with the marker in the center of the upside-down plate. While working with a dissecting microscope, transfer worms to the center of the assay plate and set a timer for 30 minutes. Put the lid back on the plate and set it aside. Continue transferring worms until all strains are on the designated assay plates. After 30 minutes, begin scoring the first plate by removing the lid and placing the plate face down under the dissecting microscope. Adjust the microscope focus until all worms are visible through the agar. Using a different colored felt tip pen from the center point, put a small dot at the location of each worm. Check the edge of the plate, as some worms may end up there. Also, count and record how many worms did not move from the center point. Measure the distance from the center point to the final location markings for each worm, and record the distance using a ruler. The first worm point is marked